This is a biology praxis prep presentation related to cellular energy. In the presentation, first we'll do a cellular energy summary, and then we'll look at some practice questions that are what you might see or similar to what you might see on your biology praxis exam. Cellular energy. Enzymes are biological catalysts. Enzymes are catalysts made by living things and are almost always proteins. Enzymes lower activation energies and speed up biochemical reactions. Enzymes speed up biochemical reactions by providing a space called an active site for the substrates or reactants to bond. This decreases the activation energy required for the reaction by putting the substrates in the right position to have their bonds broken and then remade again to form the products. Keeping the substrates close together enables them to react quicker than if they were just randomly drifting through the environment. Induced fit. Each enzyme has its own unique shape, so each enzyme is specific to certain substrates. As the substrates enter the active site, labeled A1, here's the active site, the enzyme's shape changes just a little in order to create a better fit for the two substrates here labeled B1 and B2. This is known as the induced fit model. The lock and key model states that the substrates fit perfectly without modification into the active site, but the induced fit model is more accurate as the enzyme does adjust its shape. Cofactors. Sometimes enzymes need a helper to bind at the active site to make the enzyme active. These helpers are called cofactors. Essential dietary minerals are often cofactors. If the cofactors are organic molecules, then they are called coenzymes. Vitamins are usually coenzymes. So you can see in this case, if we have an enzyme, the substrate doesn't fit directly into the enzyme. We need the coenzyme first, and then the substrate can fit, like so. Reaction kinetics. Since enzymes are proteins and protein functions are sensitive to their environments, enzymes are affected by their environments. Factors that affect enzyme activity or reaction kinetics are temperature, pH, and inhibitors, other chemicals that may be present to inhibit the activity of the enzyme. Temperature. In general, increasing the temperature of a system increases the reaction rate as the substrates are able to move faster and have more collisions with the enzyme's active sites. This is true only up to a specific temperature, the optimal temperature that you see in the graph, and is unique to each enzyme type. pH. pH is a measure of the extent to which something is acidic or basic, with 7 being neutral. pH is measured using a logarithmic scale, so a difference in a solution's hydroxyl ion concentration of a factor of 10 changes the pH by 1. The optimal pH for most enzymes ranges from 6 to 8, but is different for each type of enzyme. pH levels that vary from the optimal for a particular enzyme can cause it to change its shape, reducing its effectiveness and impact on the reaction rate. A large variance could cause the enzyme to become non-functional or denatured. Enzyme inhibitors. Certain chemicals, enzyme inhibitors, work to stop or inhibit the enzyme's function by bonding to them. Types of inhibitors. Competitive are hydrogen bonded and reversible. Non-competitive are hydrogen bonded and reversible. Irreversible are covalently bonded and non-reversible. Competitive inhibitors are similar in shape to the substrate. So you can see in the image a substrate here and a competitive inhibitor here. They're able to block the substrates from binding to the active site by binding to the active site themselves. So it's a similar shape, fits the active site, so that the substrate cannot bind. Non-competitive inhibitors bind to a separate part of the enzyme. Like you see here, what that does is causes the enzyme to change shape. When the enzyme changes shape, the substrate no longer is able to bind to the active site because the active site also changes shape and the substrate no longer fits. Irreversible inhibitors form permanent covalent bonds with an enzyme, changing its shape and inactivating it. This type of inhibitor is found in natural poisons used by organisms for defense and manufactured pesticides. Artificial inhibitors are used in medicine for destroying malfunctioning cells in organisms, i.e. chemotherapy for cancer. Ribosome-inhibiting proteins, RIPs, are irreversible inhibitors. Ricin, a protein found in castor beans, irreversibly binds to glycoproteins. When ricin binds, the ribosomes change shape and cannot synthesize new proteins, which kill the cell. Cellular locations of biochemical pathways. Biochemical pathways, also known as metabolic pathways, take place in the cell. A metabolic pathway begins with a specific molecule and ends with a product. Each step is catalyzed or sped up by a specific enzyme. Without enzymes, metabolic pathways would proceed very slowly, if at all. Glycolysis, fatty acid synthesis, and glycogen synthesis occur in the cytoplasm. Several steps of cellular respiration take place in the mitochondria. Photosynthesis takes place in chloroplasts. So a metabolic pathway starts with some starting material A, ending with some product D, and there are several reactions along the way requiring various enzymes. Photosynthesis. Photosynthesis is an anabolic pathway made up of a series of chemical reactions. The net equation that summarizes the entire process is carbon dioxide plus water in the presence of sunlight produces glucose and oxygen. Photosynthesis captures the free energy of sunlight and stores it in the glucose molecule. The free energy stored in the chemical bonds of glucose is then used to power life. Factors affecting the rate of photosynthesis: light intensity. At low levels, when light intensity is increased, you can see here, as we increase the light intensity, the rate of photosynthesis increases. At high levels, further increases in the light intensity have no effect on the rate of photosynthesis. Factors affecting the rate of photosynthesis: pH. Photosynthesis is a process mediated by different enzymes, each of which function at a different optimal pH. Rubisco, a key enzyme in photosynthesis, has an optimal pH of about 9. Changing the pH will decrease the rate of photosynthesis as Rubisco works slower. The graph shows that the enzyme works best at a pH of 8.9 right here, 100% activity. Capturing light energy. Photosynthesis depends on chlorophyll, a molecule that absorbs red and violet blue light and uses it to energize electrons to a higher energy level. The energy in this electron, which originally came from light, is stored in a molecule called NADPH. NADPH is the reduced form of NADP+. It has a hydrogen bonded to it. The electron and its energy is stored in the bond between hydrogen and NADP+. Therefore, NADPH is considered a high-energy electron carrier. This energy will be used for another part of photosynthesis. Chlorophyll is housed in thylakoids, membrane-bound structures within the chloroplasts of photosynthetic cells. Chloroplasts are organelles in plant cells and are about 5 to 10 micrometers long. Each plant cell has from 1 to 100 chloroplasts. C3 and C4 photosynthesis. There are two types of photosynthesis performed by plants to process CO2. C4 photosynthesis is about 50% more efficient than C3 and is used by maize, sorghum, and sugarcane. The C3 process is used by rice, wheat, and potato. only 3% of all plants use the C4 process. Plants in hot, dry environments use the C4 process here, where it minimizes the loss of water from the leaves. Two different parts of the leaf are involved. The light-dependent reaction, the mesophyll cell, here, is separated from the Calvin cycle that is performed in a special bundle sheath cells. Photosynthesis stages. Light-dependent reactions convert solar energy into high-energy molecules, ATP and NADPH. The Calvin cycle, light-independent, creates glucose. Light-dependent reactions. Light-dependent reactions occur in membrane-bound structures called thylakoids. The membrane separates the inside, the lumen, from the outside, the stroma, of an enclosed volume. The inside is called the lumen, the outside is called the stroma. So we have the lumen and the stroman, and we can see the light entering from the outside. And this is the light-dependent reactions here occurring in these membrane-bound structures, in these thylakoids. Photosynthesis key steps. Photosystem 2. Light energy from the sun is absorbed by the chlorophyll molecules in the chloroplast, energizing electrons. The energy splits a water molecule that releases oxygen to the atmosphere. Electron transport chain. The energized electrons move through the thylakoid membrane, transferring their energy to membrane proteins called proton pumps. Proton pumps actively transport protons, H+ or hydrogen ions, against their concentration gradient from the stroma into the lumen. Chemiosmosis. More protons in the lumen, a net positive charge, than the stroma having a negative charge. Lumen protons stopped from moving to the lumen due to the membrane. ATP synthase creates a channel for the protons. This is facilitated diffusion. The proton electric potential energy is used to add a phosphate group to ADP, creating ATP. Photosystem 1. In order to carry out the rest of photosynthesis, the electron needs to recharge as it lost most of its energy in photosystem 2. The electron reaches a second chlorophyll molecule located in a protein complex, photosystem 1, where it is re-energized from the light absorbed by the molecule. The re-energized electrons combine with NADP+ to form NADPH. Calvin cycle. The first reaction of the Calvin cycle is also called carbon fixing. Carbon in the form of CO2 enters photosynthesis in the Calvin cycle. It is turned into a solid as glucose. When glucose is used in cellular respiration, that carbon is released back into the atmosphere. The cycle uses CO2, ATP, and NADPH molecules to make a 3-carbon sugar, which is then combined with another 3-carbon sugar to make glucose. Cellular respiration. Plants use photosynthesis to convert the sun's energy, along with carbon dioxide and water, into high-energy glucose molecules and oxygen. Plants and animals use cellular respiration to break glucose down in the presence of oxygen into usable energy molecules, ATP, to power life processes. The reaction emits carbon dioxide and water. It is the opposite reaction to photosynthesis. So you can see the forward reaction of photosynthesis here and the reverse reaction of cellular respiration here. When energy is needed by cells, glucose is broken down and ATP is synthesized in cellular respiration, a catabolic pathway. Glucose can carry energy for minutes or hours and can move inside cells and larger organisms. ATP stores energy for seconds or minutes and can be used by cells in an instant. ATP can provide energy for metabolic reactions, but glucose cannot. Cells follow different paths of cellular respiration depending on the presence or absence of oxygen. Aerobic respiration takes place in the presence of oxygen, while anaerobic respiration takes place in the absence of oxygen. Here's a visual of the stages of cellular respiration. We'll discuss each of them in detail in the following slides, but refer back to this image as necessary to see the steps beginning with glycolysis, then pyruvate oxidation, the citric acid cycle, followed by oxidative phosphorylation, which is the electron transport, and chemiosmosis. The stages of respiration. Glycolysis occurs in the cell's cytoplasm, and the other stages occur in the mitochondria. Glycolysis is the first stage of cellular respiration. It involves the breakdown of glucose, a 6-carbon sugar, into 2 molecules of pyruvate, a 3-carbon sugar. The pyruvate molecules enter the mitochondrion. Pyruvate oxidation is the next step. Before entering the citric acid cycle, the 3-carbon molecule pyruvate needs to be converted to a 2-carbon molecule, acetyl-CoA. Energy from the reaction converts NAD+ to NADH, storing energy. The acetyl-CoA molecules then move into the citric acid cycle. The extra carbons from pyruvate are expelled as CO2, and the molecules of acetyl-CoA move into the citric acid cycle. The citric acid cycle, also known as the Krebs cycle, takes place in the mitochondrial matrix, the innermost space within the mitochondrion. The citric acid cycle releases stored energy by the oxidation of acetyl-CoA, producing ATP, FADH2, and CO2. The last stage of respiration is oxidative phosphorylation, which is 2 processes. The first, the electron transport chain, or ETC. The ETC involves a movement of electrons that requires the presence of oxygen. NADH and FADH2 store energy and release it in the form of high-energy electrons. If an electron acceptor, in this case oxygen, is present, the electrons will move down the membrane and transfer their energy to proton pumps. If not, the electrons won't move. The proton pumps move protons across the inner mitochondrial membrane against the proton concentration gradient. Chemiosmosis. This is the same process used in respiration, where the protons move back into the inside of the membrane via ATP synthase. The final movement of the protons uses their electric potential energy of their gradient across the membrane to create ATP. The de-energized protons and electrons are taken by the oxygen, forming water. ATP yield. The production of 32 ATP molecules assume an efficient shuttling mechanism, while 30 ATP assumes an inefficient one. These figures are lower than the previous agreed upon estimate of 36 to 38 ATP molecules from aerobic respiration. The current estimate now takes into account the energetic cost of shuttling ATP into and out of the mitochondria and the NADH made in glycolysis into the electron transport chain. So we have an output of ATP during glycolysis, pyruvate oxidation, and Krebs or citric acid cycle with a total net final ATP output of 30 to 32. Fermentation. When there is no oxygen available to act as a final electron acceptor, organisms can still break down glucose to release energy through a process called fermentation. Fermentation begins just as cellular respiration does, with glycolysis. Glycolysis results in 2 pyruvate molecules and 2 NADH molecules. But without an electron acceptor, the energy stored in these molecules can't be used. As a result, the net energy gain is just 2 ATP molecules. We use 2 ATP molecules to provide the activation energy, and 4 ATP are produced, netting 2. However, the pyruvate still needs to be cleared from the cell and the NADH converted back to NAD+ to begin another cycle. The process of doing this is called fermentation. No additional energy is released during this process. There are many types of fermentation. The two most prominent are lactic acid fermentation and ethanol fermentation. Fermentation breaks down the products of glycolysis so that glycolysis can be repeated with another glucose molecule. One glucose molecule yielded 2 ATPs, 2 pyruvates, and 2 NADHs. These are the input to the fermentation stage of anaerobic respiration. The pyruvates and NADHs are fermented into 2 NAD+ and either lactic acid via lactic acid fermentation or CO2 and ethanol via ethanol fermentation. The result of the combined steps of glycolysis and fermentation. The input is 1 glucose and 2 ATP molecules. The output is 4 ATP molecules for a net gain of 2 ATPs. In addition, lactic acid fermentation results in lactic acid, and ethanol fermentation results in ethanol and CO2. Fermentation is not as efficient as aerobic cellular respiration. For each glucose molecule, aerobic cellular respiration yields 30 to 32 ATP molecules. 55% of the energy in the glucose molecule is used to create ATP. Fermentation yields 2 ATP molecules. 3% of the energy in the glucose molecule is used to create ATP. Chemosynthesis. Deep sea vents spew hot chemicals from the Earth's dynamic crust into deep ocean waters. Photosynthesis cannot take place here as the sun's light doesn't go down that far. Organisms still need energy, which is provided by chemosynthesis, where inorganic carbon compounds are converted into organic material via oxidation reactions. Chemicals from the vents include CH4 methane, NH3 ammonia, and metals that can act as catalysts. Chemicals in the water include CO2 and H2. The energy for the reactions does not come from the sun like photosynthesis, but from the energy of the oxidation reactions. Microorganisms near the deep sea vents use these reactions to produce biomass.